Hello, this presentation or lecture deals with just one group of organic macromolecules, and that is proteins. Your study guide asks you to be able to list at least 6 different uses of proteins in the human body, other than enzymes. The most commonly mentioned function, besides the function of enzymes, is that of movement, because our muscles are composed of protein. But there are lots of other functions as well. Proteins are attached to cell membranes and have important roles in how cells communicate with one another and how they allow substances in or out of their cytoplasm. As we discuss the function of cells, probably next week, you'll become much more familiar with how proteins perform these roles, and it'll become second nature that it's proteins that are doing it. So don't feel like you need to memorize that right now. You will, though, need to be able to identify the monomers that make up proteins, just like we did for carbohydrates. As you've already learned, the name of these monomers are amino acids. And amino acids are linked together to form a polymer with bonds called peptide bonds. For this reason, sometimes proteins or the polymers of amino acids are called polypeptides because there's many peptide bonds. You need to be aware of the fact that there are 20 amino acids in the human body, and each of them differ in structure. You should also be able to recognize the names of amino acids, maybe not necessarily memorizing them all and being able to write them all down, but if you see the word of an amino acid, the name of an amino acid, you should recognize it as such. And a little clue is that almost all of the amino acids end in the letters INE, like alanine, arginine, asparagine, leucine, methionine, phenylalanine. One of these that's different that you might have heard of before is tryptophan. You might have heard of tryptophan being prevalent in turkey meat. There's really no evidence that that's true, but it's still one of those myths that hang out there. So any combination or sequence of these amino acids can be joined together to make up a protein. The ones that are joined to form a functional protein is determined and dictated by the genes in your DNA. And when we talk more about how DNA directs the synthesis of proteins, that'll make more sense to you. So let's look at the chemical structure of amino acids so that you can recognize a protein. The easiest way to do that is to look at the structure of an amino acid. All amino acids consist of a central carbon bonded to 4 different items. First, the carbon is bound to a hydrogen, and that's shown at the top of the slide in the diagram where the hydrogen is black. The second is that a central carbon is bound to a carboxyl group, and that's shown in red over here. Then the third thing is that a central carbon is bound to an amino group, NH2, and lastly to a variable group of atoms called an R group or a side chain. You should be able to diagram this general molecular structure of an amino acid, labeling the amino group and the carboxyl group. It's the R group which gives the amino acid a characteristic or chemical property that's different than other amino acids. For example, Some R groups are very polar, others are nonpolar. So some amino acids will dissolve very well in water and others won't. This ability to be dissolved in water will dictate where a protein functions best in your body and how it moves throughout the body. Is it freely suspended in the blood, or does it need some extra help? That kind of thing. So back to the generalized structure of an amino acid, we see that all amino acids to some degree are polar, regardless of the side chain, and therefore we consider proteins to be hydrophilic in general, unless there's a really big nonpolar side chain. I'd like to show you where the polar regions are in an amino acid. So this is our centralized carbon in the center here, and it's bound to a carboxyl group and an amine group. But notice that here, the way it's drawn, this amino acid has ionized. And that's because the hydrogen leaves the carboxyl group quite easily. Remember that oxygen is very electron greedy. And so the electron stays with the oxygen and a hydrogen falls off. However, the amine group is basic, and it will take up that free hydrogen because it has 2 electrons that aren't bound to anything. And so that just enables the hydrogen to bond there. So the carboxyl group is acidic. In fact, we sometimes say carboxylic acid. And the amine group is basic. So there's some polarity to amino acids that exist just naturally regardless of the R group. But I'm going to show you a few R groups, like for methionine, cysteine, tyrosine, and arginine. This is what makes them distinct from one another, these R groups. On this slide you can see that the R groups are highlighted in bright pink. Notice that sometimes an R group is really simple, like in cysteine. We just have a CH2 or a carbon bound to a sulfur. It's very simple, very small. But sometimes an amino acid's R group can be really complex, like in tyrosine. It's deceptive the way they've drawn this, but this ring contains 6 carbons. It's called a phenol ring, but so sometimes it's really complex. They really vary. You will not be recognizing the structure of individual amino acids on any assignment or test. Just like all organic macromolecules, proteins are formed by a chemical process known as dehydration synthesis. In this process, water is removed from 2 reacting amino acids. So here's amino acid 1, here's amino acid 2, and water is going to be removed. And once water is removed, then a peptide bond forms between the carbon of the carboxyl group on one amino acid and the nitrogen on the amine group or amino group on the other amino acid. So here's our peptide bond right here between the carbon and the nitrogen of 2 different amino acids. Now this peptide bond is slightly stronger than a typical single covalent bond, and that's because nitrogen has 2 electrons that aren't bound to anything, and they almost give a kind of a double bond characteristic right there. So the sequence of amino acids linked together by peptide bonds is referred to a protein's primary structure. As I mentioned, which amino acids are linked together is genetically determined. This slide shows a diagram of insulin, and that's a protein made up of 2 polypeptide chains. So one chain is on the outside and another chain is on the inside. The chains are linked, but that's not what we're looking at yet. Right now what we're looking at is that one chain on the outside has a certain sequence of amino acids. They go in a certain order. That's dictated by your DNA. Now notice that the two chains are folded. And now look at the fact that one chain has a linkage right here that contributes to the folding, and ultimately the two chains are linked together as well. So there's some interactions that are taking place between these amino acids to form a three-dimensional structure. So protein structure is a little bit more complex than just which amino acids are linked together in what order. What we just looked at was called the primary structure of a protein. But in addition, there's three other levels of protein structure. Secondary, tertiary, and quaternary are the last three. All four of these levels of protein structure contribute to the proper functioning of a protein. So we're going to look at what these different levels even mean. So first of all, we know about primary already. Determined by our genes, and it's the sequence of amino acids linked together. That's the primary structure of a protein. The secondary structure of a protein really relates to how it folds or coils. And when it coils, we use this term called an alpha helix, which I'll show you a picture of in a minute. When it folds repeatedly, kind of like a fan, a folded fan, we use the term beta pleated sheet. Beta pleated sheet. And I'll show you a picture of that too. In both cases, it's due to hydrogen bonding occurring. And so let me give you a peek at what secondary structure of a protein looks like. Notice that the top diagram has a protein. It doesn't really tell you what amino acids are there, but there's amino acids along this length. And there's an interaction between the amino acids, between hydrogen and oxygen. So they're attracted to one another, and it makes that whole chain fold. You get kind of kinks in it, if you will. That's called a beta pleated sheet. Other times, because of which amino acids are arranged in what order, you still get the same interaction, hydrogen bonding, but the folding happens differently, so that you kind of get a coil. So this is what the bottom picture is, more of a helix. It reminds me a little bit of what we see with DNA, but this is a protein, not DNA. Now let's look at tertiary structure of a protein. It causes even more folding. It's typically looser and on a larger scale than either beta pleated sheet or an alpha helix. And it occurs due to the interaction between R groups. So I'll show you a picture of that. Here's tertiary structure. And I've chosen insulin because we've already looked at insulin in the past in the primary structure. So what we can see is that within one chain of insulin, there's an interaction between two amino acids. Those two amino acids are cysteines, and they have R groups that contain sulfur. So those sulfurs are attracted to one another, and they form what's called a disulfide bridge. What's important for you is that tertiary structure is due to or occurs because R groups interact. So it's always due to R groups interacting. Always. Right there. So that's the tertiary structure. Now we'll go back to quaternary structure and look at what the heck is that? Quaternary structure of a protein always involves more than one polypeptide chain. So two chains or more are associated and linked together. Regardless of how they're linked together, they are linked together. Just the fact that for a protein to be functional, it needs to have more than one chain gives the protein a quaternary structure. And there are two examples I can share with you for that. One has to do with insulin, which we've looked at before. So this is a picture of the quaternary structure of hemoglobin. It kind of gives you a three-dimensional look of hemoglobin. This is one chain right here, and 4 chains are linked together to make the hemoglobin functional. That's kind of pretty. It looks very Irish, doesn't it? Anyway, if we look now at insulin, insulin has a quaternary structure because, again, it has 2 chains, one on the outside and one on the inside, and they are physically bonded to one another. One thing I wanted to mention about hemoglobin is that there are 4 chains, 4 polypeptides. In this particular diagram, there are 2 purple ones and 2 gold ones. And within each chain, polypeptide chain, is a heme group. I guess you can't see that very well. This is a heme group right here. And that is where oxygen binds. So one molecule or protein of hemoglobin can bind 4 oxygens because there are 4 polypeptide chains. The last topic related to protein structure deals with a process called denaturation. It's defined as the loss of protein structure, which ultimately leads to the loss of protein function. Denaturation may involve unfolding, unwinding, truncation, splitting, breaking apart. There's a lot of different levels of denaturation. One thing that causes denaturation of a protein is excessive heat. For example, when we cook meat and its texture changes, that's due to the fact that the protein's breaking down or denaturing. Another thing that causes proteins to lose their structure is altered pH. They need to operate at a specific pH to maintain their structure and their function. Protein folding is very important, and there's a set of diseases referred to, well, you've heard of maybe of mad cow disease. Maybe you've heard of mad cow disease, or maybe you've heard of chronic wasting disease in deer. Well, regardless, those diseases are due to the fact that proteins are not folded correctly. Now, it's not due to the fact that they were normal and then denatured. It's due to those diseases are due to the fact that they never folded properly to begin with. And when we do the nervous system, we'll have the time to talk about that in more detail. That's all for proteins. Thank you.